Good afternoon, dear students. Today we'll see a new topic, that is transposons or transposable elements. I hope the slide is visible to everyone. My name is Miss Anuja Kurevaru. Today we'll be seeing transposons or transposable elements. So, what are transposable elements? Transposable elements, that in simple language they mean that they can transfer from one gene to another gene. Transposable elements are also known as jumping genes, selfish genes, junk genes or junk elements. These are present in both eukaryotes and prokaryotes. In prokaryotes, these genes transport from cell's chromosome to plasmid or a phage chromosome. In eukaryotes, the gene transfers to or from the same or a different chromosomes. It causes genetic changes such as chromosome breaks or duplication. It also can cause insertional or deletion, mutation in the respective cell. Now, transposons are having insertion sequences or insertion sequence elements. These are simplest type of transposable elements found in bacterial chromosome and plasmids. And these encode only genes for mobilization and insertion. Integration of an insertional element. Now we'll see various aspects in this. Now you can see transposes recognizes the terminal repeats. These repeats IR, which is written in black and which is at the ends of the DNA. Okay, these are insertional repeats present in the insertional sequence. Then the insertion of insertional sequence into the chromosomal DNA. As you can see, in this blue part, you can clearly see that how they are straight cut in the target site. After that, insertional sequence is carried out, which you can see in the yellow part. Now, the gaps are filled by the DNA ligase enzyme. This is how a insertional sequence get introduced into a chromosomal DNA, or it integrates into a chromosomal DNA. Now, transposons have additional genes such as for those for antibiotic resistance. For example, TN3 transposon carries ampicillin resistant gene, while TN10 transposon carries tetracycline resistant gene. Insertional sequence and inverted repeats are present in the transposon, and antibiotic resistance gene is also present there. Barbara McClintock was the first scientist which received the Nobel Prize in 1983 for her discovery in the transposons in the corn, maize or kernel seeds. Kernel color alleles were unstable and they jumped from one gene to another gene, one chromosome to another chromosome, and this is what discovered by the Barbara McClintock, which introduced the molecular biologists to the new topic that is transposons. Now we'll see how transposons affect on the corn kernel color. Now you can see AC can make transposon, DS can move, but it lacks the enzyme. Two transposable elements are present at the different sites, and how they shuffle themselves together to form a different color. Generally, what we have seen, that corn is yellowish in color, but when this happens, when AC activates DS, then there is the element that inserts into color gene and inactivates it. Hence, the color gene will be purple. How transposons are replicate? First of all, they'll integrate into the donor plasmid and then they form a co-integration with the same with the help of transposase enzyme. Then the DNA synthesis will occur and the resolution of co-integrated plasmid, donor plasmid and target plasmid will occur, which will be in the original as well as new TN3 transposon. Thank you so much. See you in the next lecture.